Hi everyone, welcome. I'm Jeannie Whiting, President and co-founder of the Desmoid Tumor Research Foundation. Just a couple of announcements on this second webinar. If you weren't in our first webinar, I'll repeat that the video presentations, the recording of this meeting will be available after. You will have access to them and can watch them over and over if you desire. And wait for us, we will email you with the link to that. And also any questions that you might have, our panelists have graciously agreed to answer during and after the meeting. We will have very limited time in this meeting to answer questions, but you're welcome to put them in writing on the Q&A. You'll see for most of you, if you're on a computer, it will be at the bottom of your screen. It says Q&A, and you just type your question in. And if we don't have time to get to it during the period of time we have today, we will be sending an email to everyone who has attended, not specific answers to your specific, but a general answer from our panelists in a bulk. Also, the chat is closed for this meeting. We had over 300 people registered, patients and caregivers registered from 20 different countries attending. And we also have invited all of our doctors and researchers from our workshop and others from around the world. So our group is very large, but we welcome you all. We love this virtual format this year that allows us all to tune in. So I'll ask the presenters to please keep your presentations to the 7-minute requested limit. It is a live presentation, and then we'll have 7 minutes to answer questions. Please keep your questions to the presenter that is presenting. That will help him or her scroll through the questions at the time, during the time of their presentation or just after, and answer what they have time to answer. Thank you very much for attending, and let's turn the time over to our first speaker, it's Eldad Elnekave. No, I'm sorry, it's Dr. Bucknor. Thank you.
Okay, thanks very much. I'm going to go ahead and share my screen to get to my talk. And please, of course, let me know if anyone has any trouble viewing anything. It's a real honor to be here to present today and to talk to you, all of you, about this exciting, relatively new approach to treating patients with desmoid tumors, known as HiFU, high intensity focused ultrasound, or magnetic resonance guided focused ultrasound. I'm going to try to get through my opening remarks fairly quickly so I can address as many of your questions as I can. Nothing to disclose. So I'll just talk a little bit of brief background about the technology and talk a little bit about the desmoid tumor treatments and how focused ultrasound offers benefit to patients with desmoid tumors. And then talk about some new frontiers, thinking about, in particular, the immune response effects that we think might be associated with focused ultrasound and how that might be something we're thinking about going forward. So this technology goes by many names. If you've ever tried to Google it or search it on the web, focused ultrasound surgery, high intensity focused ultrasound, and all those describe the same fundamental technology of a high energy ultrasound transducer, same basic technology as we use for diagnostic imaging, but just increasing the energy coming out of it and focusing that energy at a single point to get that single point really hot. Now, that technology can be guided by either MRI or ultrasound, depending on the specific indication or institutional practice, although for most of the desmoid tumor treatments in the United States, at least, we do MRI-guided focused ultrasound. The basic analogy is that it's a lot like a magnifying glass focusing sunlight to generate heat and burn a leaf. You can do the same thing with sound waves coming out of a transducer, a phased array transducer embedded inside of an MRI table, producing these focused sound waves and generating heat up to 75 to 85 degrees Celsius in 20 seconds. So it gets really hot in a relatively small point very quickly. This is the device that we use for our treatments at UCSF and is used at several other centers as well. You can see in many ways it looks like a standard MRI table, except it's got this very large power cord that's going centrally to supply the ultrasound transducer that lives underneath this translucent window. So this device was originally designed to treat women with uterine fibroids. So you can see that we essentially need to bring whatever we're treating wherever the desmoid tumor is in the patient, so it's sitting on top of that area of the table. This is just a quick snapshot of one of the workstation screens I get as we're doing the treatment. So we get nice real-time feedback that we're of exactly where we're targeting, and we can see where the heat is collecting with some MRI thermometry type sequences is what we call them. The benefits of the technology are that it's completely non-invasive. There's no ionizing radiation. You can target things pretty precisely and potentially repeat it. There's no inherent dose toxicity. The key risks are that there's a risk of a skin burn, and that's probably the thing that we worry about the most with soft tissue tumor treatments. Both concerned about protecting the near field skin where the sound energy is going into the patient, and then the far field skin where a little bit of sound energy that is not absorbed may reflect back and potentially injure the skin beyond the region of the tumor. So we have to take steps to protect that skin as well. Thermal damage to adjacent structures. So really the degree of risk for the treatment is going to be driven by exactly how big the tumor is and what is near the tumor. So if it's near something that's really critical, that might be a contraindication to prevent us from doing the treatment or allowing us to only partially treat the tumor. I'll show an example of that. And then pain. Pain's not really so much of a risk, but usually patients have a few days of moderate pain following the treatment. That's easily controlled with medication. So just an overview of the clinical applications. I mentioned that the device that we used was initially developed to treat women with uterine fibroids, and it was FDA approved in 2004. There's an FDA approval for palliation of bone metastases in 2012. There's a different device. It looks a little bit like a hair dryer that was FDA approved to treat essential tremor, targeting spots in the brain in 2016. And then, those are the major MR guided focused ultrasound FDA approved indications. So we do other treatments, off-label indications of benign bone tumors, soft tissue tumors, but these are not FDA approved. We're simply able to treat patients within the scope of our practice when we think that it's a good option for patients. So on to desmoid tumors. I'm not gonna talk too much about the background of desmoid tumors because I know all of you are familiar, but I do want to speak a little bit about why does surgery fail and why might focused ultrasound offer some benefit. So one of the challenges I think with surgery is that when we have a desmoid tumor like this, so this is a cross-sectional image at the level of the hip. Here's the ball and socket joint of the hip, so the femur and the acetabulum. This is the intrapelvic area, and we're sort of slicing through the body in this way, a transverse or axial slice. This patient has a desmoid tumor just beneath the skin at the lateral aspect of the hip, sort of superficial to the gluteal musculature. And you can see that a surgeon might not have much time identifying this mass, dominant mass, if they were to operate. But I think what's so hard with these tumors is that there's often, almost always, this very infiltrative component that extends and can be very, very difficult to identify, I think, macroscopically. So that's the part that can be hard to see surgically, and which MR-guided interventions can really excel at targeting and treating. So here's an example of a large desmoid tumor that we've previously treated in this young pediatric patient. You can see it's a large tumor in the anterior compartment of the thigh. We've given contrast in this image. So the contrast tells us where the tumor is viewable, essentially homogeneously throughout the tumor. And we can see that immediately following the focused ultrasound treatment, that area, all these areas that were bright, indicating that they were viewable and alive, are now dark. So we have extensive non-enhancement throughout the tumor. This is just another cross-sectional image. You can see this is the actual transducer inside of the MRI table. We have a gel pad that helps to make sure the sound energy goes into the patient smoothly. And then there's the tumor that we saw that, again, used to be totally bright, meaning it was all live, and now is very essentially dark over a series of images. So this can be a really effective treatment in several cases. Another great thing with this technology is it can be fairly precise. So here we have a tumor that is encasing the sciatic nerve. It's the largest nerve in the body. It's not a nerve you want to damage, and surgeons obviously are reluctant to operate in this situation. But what we can do here is not necessarily totally ablate the tumor, but we can offer partial ablation, sparing as much as we can while providing a pretty good ablation of the posterior aspect of the tumor in this case, allowing for some symptomatic improvement and reduction in tumor burden. And we can bring back the patient periodically and treat them over time in that way. This is one of the larger studies that we have, retrospective studies for desmoid tumors, not a huge number of patients enrolled, but what we did see is that there was significant tumor volume decrease, 63%, and improvement in pain scores with focused ultrasound treatments. Now, it's important to talk about the limitations of this technology. For one, the cases can be quite long, 4 to 8 hours, and that has some challenges in terms of the anesthesia. Often complete ablation is not entirely possible depending on exactly where it is, and if there's a key structure next to it that's going to limit our ability to really go aggressively at every aspect of the tumor. I think focused ultrasound works best when it's paired with concurrent medical therapy. I almost think the medical therapy can do a great job at just slowly bringing down the size, or at the very least stabilizing the mass, and focused ultrasound can kind of whack out big parts of it over time, allowing us to help to bring the overall tumor volume burden, or burden of tumor volume, under control. And while it's difficult to completely eradicate these tumors, focused ultrasound offers the possibility of sort of managing them as a chronic disease with steady improvement over time. Now, another important limitation is that there's limited access. There aren't that many centers that are able to offer this, and that's really important. UCSF, where I work, is one of the centers, Stanford, Children's National, but not many others in the United States. Here's a list of, I found on this great website the Focused Ultrasound Foundation has, that sort of lists comprehensively internationally where these tumors are treated with a focused ultrasound. You can see the list is not that long. So that's definitely a place where we need to try to expand. I just want to spend just a few last slides talking about immunotherapy, because I think it is very exciting for the field of focused ultrasound. When I say immunotherapy, or when I'm talking about immune response effects, it's the fact that in addition to this ablative property of the focused ultrasound, where we can burn tissue and destroy it that way, there are other mechanical properties of this technology as well. So you can have mild hyperthermia or mechanical destruction, sort of ripping apart cells as opposed to thermally fixing them. And each of these different properties can be selected for based on the parameters of the focused ultrasound. And each of them can have different effects that can potentiate the immune response system. And that's been seen over a course of a number of preclinical studies. So there's a lot of excitement in the focused ultrasound community about how we can use this in combination with immunotherapies or other medical therapies and anti-neoplastic agents to try to promote synergy in the response to tumor treatments. I just want to leave you with one last personal example, one anecdote of why I have hope for that approach, and that's that this is 25-year-old woman with a history of a left flank desmoid tumor that you can see there. And we treated it, there's the pre-treatment appearance. You can see immediately after the treatment, we didn't have quite that solid area of darkness within the tumor, so it didn't seem like it was a total success. But over time, we see that the margin of the tumor becomes fuzzier and fuzzier. And by a year or 2 years out, we can see the tumor is just sort of slowly fading away. And that's a really unique response. I've actually never seen this response ever again. I think it really suggests the need for more research to understand how focused ultrasound might help the body to activate its immune response in the presence of these types of tumors. So in summary, this is a very effective new treatment for desmoid tumors. It works best, I think, as part of a combination approach for controlling chronic disease burden, and research into the immune response effects is an exciting new frontier, I think, for the field. So with that, I'm happy to answer any questions that anyone has. I'm gonna jump out of my PowerPoint. Yes.
Can you hear me? Something's happened with my video. If you could just please describe again the availability of this worldwide. We have a worldwide audience here.
Yeah.
How they could get more information on availability. Also coverage by insurance. I saw those questions come up.
Sure, yeah. So worldwide availability. So this is the list of sites that that I am aware of that offer this technology. So feel free to go ahead and screenshot that, or you can go to this website at the Focused Ultrasound Foundation and click through to the technology and treatment sites. So you can see just 3 sites listed in the United States. I think there is a site in the Midwest, maybe in Kansas City, that's going to come online soon, but not quite available there yet. So really not many places where it's available. In terms of insurance coverage, it's pretty hit or miss. I'd say we were successful at getting insurance coverage around in 70% of cases, but it's not something that, for example, Medicaid covers very consistently. So that is a challenge, but better than for some other focused ultrasound indications for sure. So let me.
How about the safety of HiFU compared to cryoablation?
I think that the safety profiles of the treatments are fairly similar. The biggest difference is that focused ultrasound is totally non-invasive, right? So any of the risks that are related to puncture with the needle device and the probes, so the risk of bleeding, the risk of infection, won't be present for focused ultrasound ablations. So I think that really comes into play in terms of the size and what the tumor is near, whether or not accessing the tumor with a percutaneous needle device is that much riskier than treating it with focused ultrasound. But I think focused ultrasound does have the advantage of being totally non-invasive.
Dr. Casper says we need a study on this.
Yeah, no, I think that would be great. We would love to be able to support more research into this technology. I know that there are some sites that are enrolling desmoid tumors as part of larger studies with regard to sarcoma, but I would love to see a desmoid tumor specific study because I think it is a little bit different.
Okay, and one final question. Is the treatment applicable for intra-abdominal desmoids, mesenteric?
Yeah, I would say right now, in my opinion, it's not. I don't think it's ready to go there. There are some researchers, there's a investigator in Europe who's developing this focused ultrasound to treat pancreatic lesions. And I think if we're able to overcome some of the limitations in terms of the motion of the abdomen during treatments, as well as the risk to the bowel, there's a significant risk that you could burn the bowel during the course of these treatments. If those can be overcome, it might be possible, but I think it's not quite there yet for intra-abdominal desmoid tumors.
Okay, thank you. If you have any other questions, please put them in the Q&A. And to keep on time here, we'll move on to Dr. Eldad Elnekave, who's going to talk about intra-arterial doxorubicin treatments. Eldad, are you there?
I'm here. Now, can you see my screen?
We can see your screen, but not your slides yet.
Okay. How's
there
that?
they are. Now we can hear you, so you're good to go.
Okay, excellent. All right, so there we go. I'm going to be speaking about targeted intra-arterial doxorubicin for desmoid tumors. And briefly, I have no financial disclosures. Okay, so the overview, the goals for my presentation, I want to make sure that we describe what doxorubicin-eluting treatment is as a therapeutic option, share our experience over the last 6 years in using this approach for desmoid patients, and then practically talk about how you or your patient can get assessed for this option. So we published about 2 years ago with our initial results. This was for 4 patients, all pediatric patients, and all extra-abdominal tumors. And so then we considered this as a promising approach for perplexing disease. And it's perplexing because, as we know, desmoids are local diseases, but they don't really respond well to the classic response to therapy for local disease, which is surgery. And then systemic options are varied, but generally suboptimal as of yet. Now, one promising therapeutic option systemically has been doxorubicin. It's a potent drug which is used commonly for other soft tissue diseases, sarcomas, fibrosarcoma, and has some promise for desmoids, but it's very toxic. And so the systemic administration, meaning you give it everywhere in the body, has real limitations in terms of dose, which mean limitations in terms of the efficacy for the target tissue. Now, one way to overcome that is by taking advantage of the actual composition of the drug. So Doxorubicin itself is composed of a positive, an anionic portion, which is red. That's the active part. And then a buffering or stabilizing negative portion. When you look at the solution, it's red. If you introduce small particles, these are about 100 microns, so about 10 times the size of a red blood cell. They can vary in size, but ultimately these particles can be coated in very negatively charged substance. And so as they shrink, as they sink into the solution, they actually attract the drug, and then you're left with a lot of particles that can deliver the drug. Now you can leverage that ability. So now you've got a systemic therapy which can be delivered locally if you can get it into the tumor through the blood supply. So you navigate through the arteries to find the actual vessels which supply the tumor. And here you're also leveraging the inherent hypervascularity of desmoids. Often they create their own blood supply, as is the case with many tumors. I'm going to give a couple examples and share our experience. So this was one of our first
patient to see.
Oh, one of our first patients. And this was a 6-year-old girl with, as you can see, a very large axillary tumor. And what you can identify on this slide is catheters is the main kind of the subclavian artery, the axillary artery, and then selecting vessels. Let me just make sure everyone sees this. The large tumor in the axilla, this is the T1 weighted MRI and coronal view. You can see collapse of the right hemithorax with pressing of the lung. And then what you see in black here are vessels, contrast material from the angiography. You can select the vessels which go into the tumor and then deposit these beads, achieving a very high dose of the drug locally with a very low exposure of the drug systemically. And so after 4 treatments and 1 year, what we see here is a remarkable shrinkage of the tumor. In this case, it was a 97% reduction in volume and a lot of reddish discoloration of the skin, which we often see in these cases.
Our first.
Anyway, after those first 4 cases, we saw reduction of volume between 52% and 97% in that pediatric population. We expanded the trial to include adults and to include tumors which were both extra-abdominal and also intra-abdominal, and for that matter, anywhere. So patients with desmoids in the mediastinum were treated as well. I'll show a couple of examples. This is a 17-year-old who was treated over the course of 1 year. He was given 4 treatments. And so you can see this large forearm desmoid. And then what we have in the middle is the angiographic image, selection of the intraosseous branch of the artery supplying the forearm, where we could deposit the beads with doxorubicin. And on last follow-up here, we have really kind of reduction of the tumor, some reddish discoloration, as we see often with doxorubicin, but a good response. Another patient, this is a 27-year-old with a neck-based desmoid. And we can see just a photograph on the left. On the right, you have catheterization of the ascending cervical artery. And we can see that those vessels which are supplying the mass and that kind of classic appearance for an interventionalist of a blush supplying a tumor. So treatment from that one position, there were several others. But a follow-up view here, 9 months later, we have almost symmetry of the right and left side of the neck, and the appearance on MRI, on a T2 weighted MRI, is similar. So the T2 weighted MRIs is really critical from our perspective in terms of identifying both the size, the volume, but also the cellularity of the tumor and how well it's responded to your treatment. Another example here of an intramesenteric mass. This is a 51-year-old woman. So what we have here is the pubic symphysis, and above that, a little bit of bladder. This is bowel, and then really almost most of the abdominal cavity is occupied by this desmoid. And the growth here was remarkable. So over the course of 4 months, it grew from a relatively small mass to just this enormous mass. And she was, at this point, really having difficulty just eating. What it looks like when you target these vessels in the mesentery is something like this. So you've got catheterization of the superior mesenteric artery here. A lot of these vessels are supplying small bowel and then the ileocecal, so the ileum, and then colon over here. What I've demonstrated in red here is the actual catheter. So the catheter curves up and then it enters, and this blue part is really where we want to go because this is what the suspected tumor supply was. And we can see what that looks like here. So now you've selected just that branch that seems to be supplying the tumor. We can confirm that on the angiography table using a CT to make sure that we're not targeting bowel or anything else that can be injured. And that's that's the key consideration in these procedures. This is the follow-up 10 months later. So really a reduction of almost 90% in the volume of the tumor. We treated her twice and she was hospitalized for 1 day each treatment with a total dose of about 150 milligrams total into the beads. So that's the view on coronal. On the axial view, what you see here is the kidneys in the back. So the spine in the back, belly button in the front. This is the tumor. And then 10 months later, almost a flat belly. And really, you can also see kind of the increase in fat that's deposited over that interval. And she looks much healthier. So our preliminary results with 18 patients treated with a median follow-up of 27 months, 6 kids, 12 adults, median age 37, limb, chest, wall, and abdominal wall. Those were 10 patients, mediastinal, pelvic, and intra-abdominal or mesenteric desmoids. Those were 8. More on our preliminary results. So 94% have a 2-year progression-free survival. 1 patient progressed. 89% decreased volume at first follow-up. And the median volume decrease at the first follow-up, which is around 3 months, is 52%. Median volume decrease by the last follow-up with a median follow-up of 2.3 years, 74%. Our median treatments per patient is 3. And they usually spend about 1 day in admission, follow-up after treatment. 17 of 18 patients completed therapy without a significant adverse event. So without a grade 3 or grade 4 adverse event. 1 patient had a vascular dissection, which required rehabilitation. And those are our preliminary results. And now importantly, how can you find treatment here? Well, this requires consultation with an interventional radiologist, someone who is minded to interventional oncology and delivering therapies to tumors. Doxorubicin-eluting particles are widely available. There are several different types of them. We, in this study, have used really all 3. And we've discussed and advised doxorubicin-eluting embolization for desmoid patients across the globe. So we know it's going on. Again, consultation with an interventional radiologist, an oncologist to team up. If there are questions, they can contact us for a little bit of guidance or questions. And that's the end of my presentation. Thank you.
We'll let you scroll through the questions. Could you just please go over the availability worldwide of this?
So, there's nothing proprietary about the drug or the particles. The challenge is just finding a team that's minded to interventional oncology as a procedure. Interventional radiologists who do a significant amount of work treating tumors. And that really is available worldwide. I would say most academic centers will have someone who's dedicated to this. Let me look at the Q&A. Is this the same thing as Doxil? Doxil is a little bit different. Doxil takes advantage of doxorubicin and it coats doxorubicin in a liposome. Doxil is still administered systemically throughout the body, but it is more heavily targeted towards tumors because of basically inflammation within the tumor, and it has a lower toxicity profile. But that it's different from Doxil. Risks associated with this method. The main risks here are procedural. So this isn't surgery, but it is more interventional. meaning the risks are very dependent on anatomically what you're treating. If you're treating something in the arm, there could be a risk that particles will go into the hand. If you are treating something in the abdomen or the neck, the risks are quite different. So the risks are very dependent on, really.
Could you just repeat that last part? You blipped out there. We lost your stream for a couple seconds.
Okay. Yes, and I can't see you guys, but the risks are very dependent on the case. They're procedural. This is not a pill that you take, but if it's so again, there are situations where it's lower risk. I would say extremity lesions tend to be lower risk. Mesenteric and mediastinal masses, higher risk. It just requires more diligent planning, preparation, and precautions during the procedure.
Okay, maybe one more question, please.
Marlene, I can't actually see the screen for some reason. So is there another question that can be posed to me verbally?
Jeannie, okay. Jeannie, do you want to ask? Okay, I'll ask a question.
Let's see here. Okay, now we're back.
Okay.
What are the side effects? I think so. This is, how did I just say? Okay, so the question is, can this be performed during pregnancy if the tumor is growing? So I don't think pregnancy is a contraindication. I haven't shared all of our pharmacokinetic data because it's too early, but from all the evidence we have so far, the systemic exposure of doxorubicin is minimal. I would still, I'd be cautious because there can be transmission of doxorubicin through the placenta. And I would say it depends on the size of the tumor and how much doxorubicin we would need to give. Judgment call, and it's not an absolute contraindication for us.
Okay, thank you so much. You can keep posting questions in the chat. Again, as I said at the beginning, our presenters are willing to answer questions in writing, and those will be sent in bulk to those who are registered here. We'll turn next to our next speaker, Dr. Mary Smith with SpringWorks Therapeutics, who's going to discuss the phase 3 Nirogacestat trial.
Hi, can everybody hear me okay?
Yep, you're fine.
That's good,
Okay.
we can hear you.
All right, so I have to first apologize. I'm not able to share my slides. We had a little bit of a technical issue. So I'm going to actually be giving my talk without slides. So apologies ahead of time. We'll make sure that these get posted and I can address any questions. So first off, I'd like to thank the DTRF for inviting me to participate in the panel discussion today. I wish we could all be together in person, but I'm very grateful that this meeting is able to occur virtually, and even more thankful that I'm actually able to give this talk even though I'm on my phone right now. So a little bit about SpringWorks. We're a biotech company that's advancing a diversified pipeline of targeted oncology programs, both as monotherapies or in combination with other agents in some cases. Importantly for today's discussion, I'll give you an update on our ongoing DeFi phase 3 trial, which is investigating Nirogacestat in adults with progressing desmoid tumors. And as you heard earlier from Drs. Weiss and Gounder, we just announced a collaboration with the Children's Oncology Group to study Nirogacestat in pediatric and adult patients, and that trial is activated. So what am I going to talk about today? So we'll discuss a little bit about Nirogacestat and how it works and what it is. Discuss a little bit about the results from our early phase 1 and 2 studies that led to our phase 3 study. And then of course, talk a little bit about the DeFi trial and where we are in that. So Nirogacestat is an investigational oral selective reversible small molecule. It's an inhibitor of gamma secretase. So what is gamma secretase? And apologies again, I don't have my nice pictures, but gamma secretase is an integral membrane protein that cleaves multiple other transmembrane proteins, one of which is Notch. And specifically, gamma secretase is able to cleave Notch. Once the Notch receptor has been engaged by its ligand, the gamma secretase will cleave Notch, and it releases what's called the Notch intracellular domain. And that's a transcription factor, and once it gets to the nucleus, it actually leads to modulation of gene expression for certain target genes, one of which is HES1, for example. This can, in the case of tumors, for example, that have these activating mutations, it can actually lead to proliferation, uncontrolled proliferation of tumors, for example. So blocking the Notch signaling pathway using a gamma secretase inhibitor like Nirogacestat actually will inhibit that, that whole mechanism from occurring, and subsequently the downstream overexpression of some of these genes, and will hopefully arrest the tumor. As you're aware, and you've heard in other presentations, the majority of desmoid tumors arise spontaneously and are associated with mutations in beta-catenin, or in the case of FAP, they can result from mutations in the APC gene. Both of these, both beta-catenin and APC, are part of the Wnt signaling pathway, and mutations in either one lead to stabilization of the beta-catenin protein, which leads to accumulation of beta-catenin and activation of target genes, which may underlie the desmoid biology. Inhibition, again, of the Notch pathway is thought to have an effect on the Wnt pathway because there is a hypothesis that there's crosstalk between these two pathways. In other words, that at that HES1 gene, that inhibiting Notch1 may actually play a role in the effects of beta-catenin and driving the desmoid tumor. So because aberrant Notch signaling has been implicated in a number of different tumor types, the very first study that was conducted with Nirogacestat was conducted in a solid tumor all-comer study. And this was really a study, it was a phase 1 study to evaluate the dose, to find out what was the best dose to use, and also to determine the safety of Nirogacestat, again, in this solid tumor all-comer study. There was a number of different tumor types that were studied in that study, colon, breast, thyroid, lung, etc. But importantly, there was a subset of patients that had desmoid tumors that were included in this study. What I would show you, if I could, is that all of these desmoid patients, there were 7 patients that had evaluable tumors, and all of those tumors actually shrunk on Nirogacestat. Importantly, the tumor shrinkage was actually maintained for an extended period of time. In fact, years in some cases. There was a variety of different doses that were explored in that study. The maximum tolerated dose in all of the tumor types that were tested was 220 milligrams, and the phase 2 dose that we carried forward was 150 milligrams dosed twice a day. Again, there was about 70% of patients that responded in that study. That led to a specific study that was conducted by the National Cancer Institute. It was a phase 2 study that actually evaluated both the safety and efficacy of Nirogacestat. There were 17 patients that were enrolled. And again, this was a patient population that had progressing desmoid tumors, and it was a very heavily pretreated patient population. So these patients were considered refractory. They had failed anywhere between 1 and 9 lines of therapy. And again, these patients were able to respond to Nirogacestat. Their tumor shrunk, and in 30% of the cases, they had a partial response. In other words, and the rest of the patients had disease stabilization. And again, this was really independent in both studies of whether or not patients had an APC mutation or they had a beta-catenin mutation. So if we look at the totality of that data and all the patients in the phase 1 and phase 2 study, we look at what the tolerability of Nirogacestat was in those patients. And again, this was probably about 70 patients. The most commonly reported adverse events are similar to those seen with other gamma secretase inhibitors and include gastrointestinal issues such as diarrhea, nausea, vomiting, hypophosphatemia, which is a decrease of phosphate in the blood, which actually can be supplemented with oral phosphate replacement. And then patients also developed rash. So where are we right now with our DeFi study? Most importantly, I think you heard from some of the previous talks that the DeFi trial has completely enrolled. So I'm really happy to say that. Where we are right now is that you know, we had a target enrollment of about 115 patients, and we met that target enrollment this summer. We had about 50 sites that conducted this study, and this was across the US, Canada, and the EU. So again, a shout out to everybody there. The study was designed as follows. It's it's a double-blind placebo-controlled study. The patient population that was included for participation had progressing desmoid tumors. Patients were randomized 1-to-1, and there was an open label, or is an open label extension for patients who progress on study. The primary endpoint for the study is progression-free survival, very similar to the sorafenib study that has been reported out previously. And we have a number of key secondary endpoints that we're looking at. Of course, safety and tolerability, but also what is the objective response in these patients? What's the duration of response? And as Dr. Gounder has also mentioned in his talk previously, through the development and the collaboration of the DTRF and Memorial Sloan Kettering, they developed a desmoid specific tool that we are able to use in our study to help validate that tool so we can see how well we are able to, or Nirogacestat is able to affect the quality of life in patients. Again, we started this study in the first half of 2019, and I'm happy to say we are fully enrolled today. So what does that mean right now? So what happens in this type of study is that we're in what's called the maintenance phase. So we patients will continue to be evaluated for tumor responses and will be assessed. And once we reach a targeted number of events, then we would be able to analyze the study. So we're basically in that maintenance mode right now. So that sort of concludes my discussion, but I just want to actually say a very heartfelt thank you to all of the patients and the investigators who were involved in the DeFi study and are still involved today. And a special thank you to the DTRF for not only their tireless advocacy here, but again, also for all of the work that they do for the desmoid community. So thank you very much. Again, I'm very sorry that you just had to look at me during this talk and I didn't have my slides, but you know, we'll make sure those get posted.
Thank you, Dr. Smith. That was a great presentation. Am I correct that you cannot see the chat, the Q&A?
Hold on one second. I am able to, I see a list.
Can I just ask, pass on one question I'd like you to start with, then you can scroll through.
Sure.
Availability to patients now that the study is full to this
So
drug,
right,
whether compassionate
yeah,
use or otherwise.
that is correct. So we do have a compassionate use program. You can go to our website, which actually, well, let me just back up for 1 second. If there is a patient who is interested in Nirogacestat, they would have to discuss this with their treating physician. The treating physician would then make a query to SpringWorks through the website. There is a link there, requesting access to Nirogacestat. And again, it really just depends on the situation. Each case is evaluated on a case-by-case basis. So that's how you would be able to access it, but it's through your treating physician. They would have to request it. Let's see, what are the locations of the majority of tumors you are speaking to? So in our study, we did not limit the location of the tumor. So again, this is a blinded study, just as a reminder. We enrolled patients that have extremity tumors, intra-abdominal tumors, but we did not have any restriction on mutational status. So patients that had FAP were able to be included in the study. Okay, let's see. So compassionate access we talked about. So again, I encourage everybody to go and talk to their investigator. Let's see. Let's see. Then could you please comment on hypothesis? Let's see. So so side effects are so somebody had said that the side effects are intolerable, but the tumor has shrunk. So again, what I would suggest that again, it's a blinded study. If there is a patient who is out there that is on the study that needs to discuss their adverse event profile with their investigator, I would suggest that they do that specifically with their treating physician, because there are some things that can be done, for example, a dose reduction depending on the adverse event, etc. Let's see, several patients that had stress fractures due to bone density loss. I will have to get back to you on this. Talking about stress fractures in the phase 2. I'm not familiar with that specifically, so I'll have to go back and look at that. As a reminder, that was a study that was conducted by the National Cancer Institute. That's actually I don't want to say it's ongoing. There are patients that are actually still continuing in that study. In terms of the information that we have, we are in discussions with the National Cancer Institute to obviously get the full breadth of the data from that study. So again, there are still patients that are ongoing on that study. I believe it's 5 still, 6 years later.
Okay, we can take one more question, Mary.
Yes, let me just, sorry, in order to look, I have to go back and forth, so excuse me here. The side effects, I think I talked a little bit about those. Again, we've seen a number of side effects in the phase 1 and phase 2 study. Let me pull them up. Again, the most common events that we do see are those gastrointestinal. This is actually a very common side effect with other gamma secretase inhibitors. So the nausea, vomiting, and diarrhea is actually one of the most common adverse events. And again, if this is something that is being observed, you should talk to your investigator about that to seek some treatment for that. In addition to that, rash was observed. I mentioned the hypophosphatemia, and sometimes you can also get other side effects with some of your blood levels and things like that. So,
and there are several questions about early menopause or fertility, future fertility concerns.
Yes, so obviously the study is ongoing, so any kind of long-term effects on those types of outcomes are still being evaluated as part of the study. Again, it's a blinded study, and we continue to evaluate all adverse events so that we can fully characterize them and understand, for example, when they occur, and if they are, any of these events are reversible upon stopping treatment. So all of that information is being gathered as part of this study so that we can more accurately be able to assess each of the adverse events.
Okay, thank you very much. And again, if there are
Sure.
any other questions, you're free to put them in the Q&A. Thank you. We'll
Yeah.
move on to our final speaker, Dr. Cunningham.
Yes, thank you.
Iterion Therapeutics.
Okay, thanks. Welcome everybody. And of course, I want to add my thanks to the DTRF. And actually, I'd like to start by congratulating Mary and the SpringWorks team. Those of us who are in the business of bringing drugs to patients know how rare and special it is to get all the way to a phase 3 trial and get that completed. And I offer complete congratulations to her and her team. So I'm going to go to share my screen now. And there. So I want to talk about a phase 1/2 trial of a drug called Tegavivint. Those of you that have been to previous DTRF meetings have heard me speak before about this trial, but for those of you that are new to this, I'm going to spend just a second to sort of give you the mechanism of action of the drug. So a common question from people with desmoid tumors is, if beta-catenin is the problem, why aren't we just drugging beta-catenin? And the answer is that over the years we've tried. The difficulty is that beta-catenin has other roles in cells besides just its actions in the nucleus. And if you globally inhibited beta-catenin function, you would also inhibit those other roles, some of which have to do with how cells stick to each other. And so you get a lot of side effects, particularly gastrointestinal side effects. So that's why most drugs that you've heard talked about have other mechanisms of action that then try and indirectly impact a nuclear beta-catenin. And Tegavivint takes that approach too, although it's a little closer to the target. So it binds to a protein called TBL1 that binds to beta-catenin in the nucleus. And so the hope was, and what the early research at the bench and preclinically showed, was that binding to TBL1 inhibited the nuclear activity of beta-catenin, but not the other roles that beta-catenin plays in the cell. So we had some idea and some hope that this would escape the toxicities that some of the other drugs in that pathway have seen. And so we decided to conduct a first-in-human trial. And I have to emphasize, as I have at previous meetings, that this is the first time that people have been given this drug. So there's going to be a lot that we don't know, and we have to proceed very carefully in these early trials. So this was an open-label trial. It was held at several centers. It was not randomized because this is, again, what we're trying to show in this trial is whether or not the drug is safe as the primary goal. And then we'll also look for evidence of activity. So it was a two-part study. In the first phase, we took small groups of patients and gave them a dose of a certain dose level of the drug, and then we watched them. And the schedule was that this is an intravenous drug, it's not oral. They got an infusion for 3 weeks in a row. It takes about 4 hours to give the infusion, followed by a week off. And after the first month, if they looked okay, we would take the next group of patients and give them a slightly higher dose of the drug and keep marching up like that until we saw some signals that said we didn't need to go any higher. Now, in most phase 1 trials, those signals are side effects. You begin to see side effects that you know are going to just increase if you give more of the drug, and so you stop. And I'll also say, and I'll repeat this later, if you want more information about this trial, clinicaltrials.gov is, of course, a great resource. So far, what have we seen? Well, we've got 19 of the
Dr.
plan.
Cunningham, some patients are saying, we can't see slides. Did you have slides or not?
Yes, I'm showing slides.
Okay, we're not seeing them.
Okay, let me see.
You share your screen, please.
Yeah, I thought I was. Let's try it again.
There you go. Now we've got it.
Okay. Can you still see it?
We can, yes.
Okay, that was where the glitch occurred before. So
Okay.
this is the slide I was talking about, the mechanism of action, and it's just the usual cartoon. And then this is some of the description that I was giving you about the trial. I was so awestruck by Mary's presentation without slides. I guess I was trying to do that too. So here's a summary of the results to date. And as I said, this is a phase 1 trial. So our primary concern is what is the drug safe and what are the side effects? So we enrolled 19 of the planned 25 patients so far. It's actually been very well tolerated. That's what we expected, but we're still relieved to report that. We've not had any toxicities that required lowering the dose or stopping the drug. What we ended up doing was saying that the sort of recommended go forward dose was based on blood levels. We saw that as we were beginning to increase the dose, at some point the blood levels just leveled off, and so giving more would not have done any more good. The other point to note is that because patients have tolerated the drug well, they've been able to stay on quite a long time. And so even though it's only a few patients in total numbers, since so many of them have been on the drug for months and months, we've got more than 160 cumulative follow-up months. And so we think that leverages the signal that we're seeing, even though the numbers are small. So what have we seen in terms of efficacy? Well, here's a swimmer's plot. Remember that, and you know this better than anybody, that desmoid tumors are pretty slow to respond, even when they do respond. So it takes a while. So far we've seen, and it's also important to note that all the patients that were enrolled had actively growing tumor symptoms related to the desmoid when they came on. So far we've seen, I hope this thing's not doing this automatically. So far that we've seen two partial responses, and a partial response under our criteria is defined as tumor shrinkage of 50%. We've got a third patient who's closing in on that, and we'll see what the next scan shows. Most people have had some tumor shrinkage, but it just qualifies as stable disease right now. We've got 9 patients remaining on treatment with stable tumors, several for greater than 1 year. And anecdotally, several patients are reporting less pain. Now, we're also going to use that patient-reported outcome tool that Mary alluded to in their trial. We'll do that as patients get on the go-forward dose. And I'll also mention that the patients in the lower dose cohorts, we did offer the option, if they were still on the trial, to increase their dose up to the one that we were going to carry forward. So here are the clinical centers that are currently enrolling patients in this trial. There's one in Toronto, the rest of the United States, Seattle and Boston, MD Anderson and Houston, Memorial, and Ohio State. So we are pleased that we've gotten this far in the trial. It's still early days yet. We've just determined the dose. We're pleased at the side effect profile. We think we're beginning to see some signs of activity. But it's, I can't emphasize enough, when you do drug development like this, you have to proceed slowly and cautiously. And because of that, there are some restrictions in who can be on the trial. And that would include, let me stop sharing the screen. And that would include, currently we're not allowing patients with FAP on, we're not allowing children on. As we get more knowledge and comfort with the drug and its activity, we may can start to relax some of those parameters. But since this was the first time the drug was going to be given to people, we wanted to be as cautious as we could. And so with that, I'll stop, and I'm going to start scrolling through the questions. Yeah.
Could you describe the difference now you've moved into phase 2A of the trial?
Yes. Yeah, so phase 2A just means that we now know the dose that we're going to carry forward. And so you could call it a phase 2 trial. So the jargon in clinical trials is phase 1 is where you're defining the dose and the side effects. Phase 2 is where you're asking the question, does the drug work in a particular kind of tumor? It's sort of been the fashion recently to do even your phase 1 trial in patients with only one kind of tumor. So you sort of combine those two. But generally a phase 2 trial, a true phase 2 trial is going to be a bigger trial. You know, it's going to be north of 40 or 50 patients. And a phase 1 is generally 25 or less.
Okay, do we have another question? So the side effect profile you've seen so far?
Yeah, so we haven't. I mean, that's there's been a sort of a list of, because we quiz people every time they come in. Are you feeling anything? What's happening? And so they have a variety of symptoms that they see, and then it's up to the investigator to decide whether that could be due to the drug or not. One way you do that is if the symptom occurs when you give the drug, and then remember I said there's that week off. We deliberately put that in there so that if we saw certain symptoms that came on and then they got better when you weren't getting the drug, then we'd know it's more likely to be the drug. So, you know, people have had the usual sorts of things that you get going through life, headaches and stomach upsets and joint pains and this and that and the other, but we haven't been able to identify anything that consistently shows up when you're getting the drug. So that's one reason why we're still studying it. There's
one question there about neuropathy. Has that become an issue?
Yeah, so there was a report or two of some patients that had some numbness and tingling in their, I think it was one patient had it in their leg at one of the centers. The investigator alerted us, and so we immediately went through the database. All this is captured electronically to see if anybody else had that. We didn't find anybody, but we're continuing to follow that pretty closely because it is something that you'd want. You know, that's the kind of side effect that occurs over time, and then it often doesn't get improve all that much when you stop the drug. You know, I go back to the fact that we've had patients on for more than a year now, so fingers crossed that that's not going to be a problem, but we're watching that pretty closely.
Okay, I think we covered most of the questions.
Yeah, most of them where they can't see the slides.
Summarize again, please. How many patients spots do you still have available?
So right now there have been 19 enrolled, and the study size was to be 25. So there should be 6 additional slots. It's possible there could be a few more if we need, if some of the earlier patients it was determined that we didn't get all the information that we needed. But that's at some point you have to go on to the next trial. So I would say there's probably about 6 slots left.
Okay, thank you so much. And again, any other questions, please put them in the Q&A in writing. It's been an incredible experience to bring people and doctors and researchers in from around the world. For us as a foundation, it's really been incredible. We thank you all for attending. I wanted to close with just a couple of announcements. Again, we thank our sponsors who have helped make the weekend meetings possible: SpringWorks Therapeutics, Iterion Therapeutics, and Ayala Pharmaceuticals. And we thank you all for attending. Again, these webinars will be accessible via our website eventually, and we will send you answers to the questions that were not answered. Just give us a little time and we'll email that out to all of you, so you'll have that information. Do any of my colleagues, I just want to close introducing Marlene Portnoy and Lynn Hernandez. The 3 of us basically run the foundation together. We're used to the virtual format because we're in 3 different states as it is, and we've worked very efficiently for many years in this format. But do either of you have anything you wanted to close with? Marlene, Lynn, you can unmute.
No, thank you. I just want to thank everybody for coming. This is our first venture, as Jeannie said, into the virtual world, having a virtual meeting, and it's been really successful. But we look forward to seeing everybody in person next year. So, but thank you for attending.
Yes, and on that score, please save the dates: Saturday and Sunday, September 25 and 26, correct?
Yeah.
We'll send the save the date in the email, but we're already prepaid in Philadelphia because we moved this year's contract into next year. So we're sure hoping we can all be there in person. Okay, thank you, everybody. Goodbye. Have a great
Quickly,
week.
I'm sorry, one more thing. So I just want to remind you guys that tomorrow we have our RFA virtual challenge ceremony, which is taking place from 11 to 11:30 a.m. Eastern. This is going to be a great opportunity for you guys to see the faces and hear the stories of other desmoid tumor patients. It's going to be uplifting. It's going to be inspiring. So we invite you to join us to close the weekend with us tomorrow.
And it has been so successful because of all of you taking the virtual challenge. You'll be so excited to hear how much we raised.
Yes.
Really, it's just going to be a fun celebration, whether you participated or not. Please sign in. It's just our fun party at the end of the weekend.
Yes, join us on the DTRF Facebook page. You'll be able to find us live.
All right, thank you, everybody. Best of luck on your desmoid journeys. Thanks to all our doctors and researchers who take care of our patients and are moving the research forward towards different and new and effective therapies and eventually a cure. Goodbye. We'll see you tomorrow.
Goodbye, everybody.